Hello students, now in this video we will understand how to tackle this lengthy types of question. Okay, so basically I'm not going to reduce the time period which is required to answer this question. Here we are not focusing on how do we solve a question when it has been asked in board exam or in any kind of entrance exams like NEET or anything else. So here first we will understand the concept, okay, what is required to answer this question, and then we will step by step clearly understand how can we answer these four sub questions. Now let us get into the explanation quickly. First, let me read the question. ELISA for tracking disease spread. Okay, yes, so this concept is about ELISA. Okay, I'll help you understand what is it. Now here there are four sub questions. Okay, first one is what happens to the proteins in plastic well if the sample contained an antigen. Okay, so this is depending upon the working principle of ELISA. Okay, what if it did not contain antigen? Again, working principle. Why are enzymes used? Okay, in the immunoassay, again working principle. Why is it important to able to detect antibodies in people who do not appear sick? Okay, this is application of ELISA. And the next question, if you have tested positive for a disease exposure but did not have direct contact with one of the original infected students, okay, then what conclusion can we make about the transmissibility of the disease? This is again about the application. Okay, now let us get into the explanation. Now, as I told you, here the concept given or what we need to understand with clarity is ELISA. ELISA, when we start, understanding the abbreviation of this is called as enzyme linked, okay, immuno, I for immuno, okay, assay. Otherwise, it is also called as immunosorbent, S for sorbent assay. Okay, so E for enzyme, L for linked, I for immuno, okay, S for sorbent, and A for assay. Sometimes the sorbent is also removed. It is called as immunoassay. Now, this is a laboratory test, okay, which is conducted to detect a disease. Okay, now here a specific kind of disease is detected depending upon the kind of specific antigen what they find in a sample or antibody, or sometimes certain types of glycoprotein. Now you know the story behind the antigen and antibody, right? Now antigens are the surface proteins, or the proteins which enters the body on infection. Now, in response to this infection or this infectious protein, antibody is being produced from the body as an immune response. Correct? Now, the antibody produced is obviously going to be antigen specific, and antigen which is entering the body is going to be the pathogenic specific. Right? So each kind of bacteria or each kind of virus has its own kind of antigen. Okay. Yes. And the antibody produced to fight it is again going to be specific. Now finding the specific antigen or antibody will help us detect what kind of disease is there or is being infecting a specific individual. Okay. Yes. So this is a laboratory test which is used to detect the disease based upon the type of specific antigen or antibody which is being found in the sample from an infected person. Okay, yes, understood? Now to answer all these, four sub questions which is revolving around the first two sub question, as I told you, is based upon the working principle of ELISA, whereas the next two sub question depends upon the application of ELISA. Now, let us understand the principle so that we can answer first two sub-question, and then based upon the working principle, you'll automatically get to know the application, which will help us answer the next two sub-question. Let us get into the principle with the help of a suitable image. Now we are talking about working principle. I'll put it as WP. Now look at in the image. Now to conduct ELISA, a plate which is also called as microtiter or ELISA plate is being taken, which has 8 in column and 12 in row. We don't count the number here. I'm not counted here, but I'm telling you, the row has 8. I'm sorry, the column has 8 and the row has 12 wells. The sense, if you look at it properly, there'll be wells like this. Now into this well, the sample is added. The sample collected from the infected individual. Now this sample can be blood, urine, etc. A fluid sample. Now as you see here, what happens? This is the well, the blue colored. The well what you see here. Yes, it is the well or the microtiter in the plate. The ELISA plate. Now, after the sample is being added, it can either contain the antigen. Now we are detecting the antigen. It can either contain the antigen or it will not always have the antigen too, because it is not important that a person whom we are suspecting is infected. We are going to find whether he is infected or not infected with a specific disease, right? Now this ELISA helping in detecting the specific antigen. Now the sample, if it has been added, if it has an antigen, what happens? Next, after adding the antigen with a specific buffer solution, so the sample is treated with a specific buffer solution. Why? Because buffer solution is going to control the reactivity of this antigen. Yes, and then a primary antibody is being added. So antibody is a protein. So a mixture of antibody is being added. Now, if that antibody, has a specific kind of interaction with the antigen in the sample, then it interacts and there'll be antigen antibody interaction, as you see it here. It is specific. Not all the antibodies will cling to that antigen. There will be interaction. Now, this kind of interaction is actually hydrophobic. Which is not a water lover. It is non-polar bond, what you find. Now, after adding the primary antibody, a secondary antibody is added. I'll put primary antibody as first degree here. A secondary antibody which is added, which is nothing but enzyme linked antibody. Now the enzyme what has been used here, the most common one is called as the reddish peroxide. So here the antibody taken is the antibody from the horse. The H stands for horse here, R stands for reddish, PO stands for peroxide. So here it is the enzyme which is nothing but a peroxide, and an antibody cling to it is the antibody from the horse. That is what is called as HRPO. Now this enzyme carrying this specific secondary antibody will have the interaction with the primary antibody if there is a specific interaction among them. Now this secondary antibody is actually added in order to compete the antigen. So wherever wherever in the wells, if you see, there has been an antigen-antibody interaction taken place, then the secondary antibody reacts or interacts with the primary antibody, which is fastened by the enzyme here. Enzyme catalyzes the interaction between the secondary and the primary antibody. Now, if that interaction would have taken place, then there is a colored change. What you see, that's what we've put here as colored product. Now, suppose the sample which I have put in green color, okay, for example. Now, after adding the enzyme, if the color changes to blue color, that means there has been antigen-antibody interaction taking place. That means a specific antibody which I have added has interacted with the specific antigen. And this is indicated by the color change after adding the secondary antibody. Now, after this, okay, a TMB substrate is been added because this one, what it does, okay, it actually stops the further reaction, because this further reaction between the antigen and antibody has to be stopped, and even between the secondary and the primary antibody has to be stopped, because there are other antibodies also been there in, the substrate what we have been adding for finding the antigen, in order to stop that reaction, like how we have the stop codons while translation has been taking place. Similar to that, specific substrate has been added in order to stop the further interaction between antigen and antibodies, right? Okay, now this is how ELISA works. Understood? Yes. So this is the working principle. Okay, now what was our first question? If you see, and knowing the working principle, we can answer the first and second sub-question. First sub-question, what happens to proteins in a plastic well if the sample contained antigen? And what if it does not, or it did not contain the antigen? Now, if the sample contained the antigen, okay, then a color change we can see, right? Because if it contained antigen, a specific antigen-antibody interaction would have taken place, okay? After adding the secondary protein here, nothing but enzyme-linked antibody, okay, this interaction would have been tracked after adding the secondary protein. There would have been color change. If there is no antigen at all, okay, there is no, binding reaction which takes place with the antibody. Even after I add the secondary protein, it won't be indicated with any kind of color change. So there won't be any color change. Understood? Yes. So we have answered the first sub-question. Next one, why are enzymes used in this immunoassay? You can answer it clearly. Enzymes are used to track the interaction between antigen and antibody in this entire process. Okay, now the antibodies which has been linked with the enzymes, they compete with the primary. They compete with the primary antibody. Now, this can actually indicate if our sample had antigen or not. One is there won't be any color change if there is no antigen. And there would have been color change if there is antigen. So enzyme, by tracking the interaction between antigen and primary antibody, it indicates the presence or absence of the antigen. Correct? That is what enzymes do in this ELISA technique. Okay? Yes, we have answered the first two sub-questions. Now let us answer the second two sub-questions. Okay? Next one is, why is it important to be able to detect antibodies in people who don't appear sick? Now just now we had gone through a pandemic called as COVID. And we had also discovered in the sequence of its effect that certain individuals also become asymptomatic. That means they are the carriers of virus or the pathogen, but they do not show any kind of symptoms. Just because they are not showing any symptoms, those individuals cannot be called that they are not sick or that they are not being affected. Now, this asymptomatic people, if they are coming in exposure to a healthy individual, they have all chances of spreading the disease, right? So that is why even if that person does not appear sick, the antibodies or the antigens produced by him when tested via ELISA, we can easily tell that if that person is positive or negative. That is nothing but positive with the disease or negative with the disease. Okay, by this we can actually control the spread of a specific disease which is persisting inside an individual but does not show any kind of symptoms. Okay, this is with specific individuals where if there is no kind of symptoms we see, but specific individuals we do see the symptoms. It is blindfolded, so we don't know who is being affected, who is not affected. So in that way, this laboratory test helps us understand who is literally affected, even if there is no symptoms seen. Okay, yes, so this answers our third sub question. So the next sub question here, what it is? If you had tested positive for disease exposure, okay, but did not have direct contact with one original infected students, okay, what conclusion can you reach about the transmissibility of the disease or of disease in a population? So similar to that, okay, suppose I have been tested positive with COVID. Let me take this scenario itself. There are other diseases also where you see asymptomatic individuals like typhoid. Okay, but I wasn't in contact with any of the infected person. So what can I conclude here is diseases like these two are transmittable. Okay, via carriers. These carriers are the living organisms or the humans or the, any organisms which can carry this specific pathogens, okay, or the infected organisms. Here it is a virus. Typhoid is caused by Salmonella typhi bacteria. So by this, we can get to know that these diseases are transmittable or communicable disease, which can spread via carriers, okay, and can also spread via mediums like air, okay. Typhoid can spread via contaminated food, water, fecal matter, etc. So what can I tag here saying that it is a communicable disease in a population? Okay, and under that there are certain diseases which passes via carriers where you cannot find any symptoms also. Okay, understood? We have answered all the 4 sub questions. Now, as a final answer, what I wanted to tell you is ELISA is a laboratory technique to detect specific antigen or antibody which is produced by an infected person, okay, on affecting by a specific disease. Now, this technique can be used to control the spread of a specific disease. Okay, yes, and enzyme linked immunosorbent assay works on the presence of a enzyme which is linked with a secondary antibody. Yes, so this entire interaction depends upon an antigen and an antibody. Yes, clear? I think you have understood the entire concept, and I hope you can answer questions like this which ask multiple questions on one specific concept itself. Thank you so much.